Okay, so today I'm going to tell you a story about two organisms and how genomics and genetics have played a role in the modifications of these organisms and for their use in agriculture. So the title of my talk is Corn and Cows, the Genetics and Genomics of Agriculture. So if I were to ask you what inspired Darwin's theory of evolution, probably you would say beak of the finch, his acquaintance with the birds of the Galapagos, which he encountered, of course, on his voyage. While that's true, of course, that's not the whole story. And in fact, Darwin was inspired as well from his deep knowledge of the process of domestication. And in fact, in The Origin of the Species, he describes what happens to organisms under the process of domestication. And he even wrote a 2-volume book entitled Animals and Plants Under Domestication, in which he described in great detail some of the kinds of changes that occur as plants and animals live in association with humans and are modified by humans. Part of the inspiration for this was, of course, his familiarity with pigeons. They were a very popular pet, if you will, at the time. And of course, breeders had spent a lot of effort generating all different kinds of pigeons. And so he was familiar with the very rapid changes that could be generated in this bird as a result of human intervention. Now, he didn't need to look at pigeons, of course. He could have looked at an even more common pet, if you will, which is, of course, the dog. Dogs are descended from wolves here, and we have, over the 10 to 14,000 years of, that we've been living with dogs, changed them and modified them in all sorts of interesting ways. Early on, we probably modified them primarily for the purposes of performing particular tasks, for example, herding or working or hauling things or what have you. But of course, more recently, we've imposed our aesthetic on these animals, and now we produce dogs that have all sorts of strange and interesting traits that, for whatever reason, humans find fascinating. So the dog is an interesting example of what kinds of changes humans can generate on the organisms that they interact with. Now, you might not be as familiar with this, but we've created similar kinds of changes in essentially everything we interact with. So when you think of sheep, you probably think of organisms like this, that is, animals that have long wool, and that wool is typically white. But in fact, that's not what sheep look like in their original wild state. These are wild sheep, and as you can see, like all wild mammals, their coat color is brown, not white, and the fur is short, not long. And the males in particular have very long horns, which the domesticated sheep do not. And so these are the kinds of genetic changes that humans have selected for over the thousands of years that we have been raising sheep for milk or for meat or for any other food source. Another, of course, wonderful example of what humans can do to organisms is shown, but this is one of my favorite examples, and of course a very popular example of showing what can be done with just a little bit of genetics. Brassica oleracea is the name of this little plant here. It's not a very interesting plant in its original state, but over the years that humans have grown it, we've identified various mutations that have had dramatic effects on what this plant can do, mutations that affect its flowers, like these. So we have cauliflower and broccoli, or kohlrabi, or Brussels sprouts, or cabbage and kale. All of these common brassicas are the same species. They are this plant, and they are essentially mutant varieties of this rather nondescript plant. Another example that you may not be familiar with, of course, is the carrot. When we think of carrots, we think of these orange, rather perfect-looking roots. But this is a modern invention. The carrot in this form, that is, the orange carrot, is only a couple of 100 years old. The wild carrot, from which this is derived, looks something like this, which is it has roots that have very low levels of beta-carotene. They're not orange, and of course are highly branched and quite irregular. So again, this modern carrot that we all take for granted is a very recent human invention of only a couple of 100 years. So when we start to think about what kinds of changes have occurred as a result of humans becoming interested in an organism and modifying that organism, we can divide those changes into two sort of general categories. The first category we refer to as domestication. And domestication is really the sort of step, the first step in the modification of an organism, where it's brought from the wild into a human habitation and modified in various ways that allows that organism to live in association with humans quite happily, and in many cases eventually become completely dependent on humans, so that the organism, whether it's a plant or an animal, could not survive in the wild. It loses its capacity to survive in the wild. And that process we term domestication. Now, once that process has occurred, once the organism, the dog, if we're talking about dogs, or corn, as I'll tell you in just a bit, becomes dependent upon humans and is grown for a long period of time, then we start to select for different varieties of that organism that we like for different reasons, that meet either our aesthetic needs or our needs for improved food production or what have you. And that process we term improvement. Although in some cases it's not improvement at all. In fact, it makes the organism less viable and less capable of actually surviving. So a good example of this, and perhaps certainly the one I'm going to focus on today, is what happens with corn. So this is a picture that John Dobley took, and John Dobley at the University of Wisconsin is the person who's really worked out the evolution of corn. And he basically showed that corn, as we know it, shown here in this sort of typical ear, evolved from a plant called teosinte, shown here, that, as you can see, produces a very small ear with very few kernels. And one of the outstanding features of those teosinte ears is that the kernels fall apart. They so-called disarticulate. And this is, of course, a very useful trait to have if you're a wild species. You want to disperse your seeds. That's what wild organisms do. But humans do not want seeds falling on the ground. They want seeds remaining in place so that they can harvest an ear, for example, and have all the seeds remain on it. So as humans selected selected this variety for as an improved food source, they ended up eventually with something like this. Now, this, of course, is not what the earliest forms of domesticated corn looked like. When the Pilgrims arrived in the New World, the Northeast, this is the kind of corn they encountered. So the first Thanksgiving, this is probably the kind of corn that was consumed at that time. This is so-called northern flint. This is a picture taken by Pat Schnabel at the University of Iowa. If you go farther south in the United States, you find this kind of corn, the so-called southern dent. And sometime in the movement of people around North America, these two varieties hybridized to produce this type of variety, which is the so-called Midwestern dent, which is the corn that we're all familiar with growing throughout the cornfields of the Midwest or Lancaster County or anywhere you encounter corn. Now, before the middle part of the 20th century, of course, farmers worked very hard in the 19th century in particular and before to, of course, grow the best variety of corn. And the way they did that was they'd go through their field, they would find a plant or an ear that was particularly large or a plant that they liked, and they would save that ear for planting in the next season, right? And they were able to, generate good varieties of corn, but highly variable varieties. For example, this is Lancaster Surecrop, which is one of the most popular varieties of so-called open-pollinated corn, grown in the 1930s. Okay, so, but it's, as you can see here, that's no self-respecting farmer today would consider that a great ear of corn. If we look at the information about corn production from 1866 to 2011, you can see that for quite a long time here, from 1866 up until about 1930s, there was no measurable change in corn production, despite all that selection that farmers were doing for big ears of corn and pretty ears of corn. So the yields of corn were pretty much flat. And then something amazing happened right around this period in the mid-30s or so. And that something amazing was hybrid corn. So right around this time, hybrids were introduced, and these were the product of the research of basic scientists. And what they did was they took corn, that sort of Lancaster Surecrop, that variable type that was common at the time, and they started self-pollinating it over and over and over again to produce so-called inbred varieties of corn, highly, highly inbred varieties, so that they're basically genetically uniform. And two very important inbreds were B73 and Missouri 17. And these are highly inbred. Again, you can take this, they're self-pollinated, and the progeny of these plants will just look just like their parents because they're completely inbred. Now these plants right here, this B73 gives you an ear that looks like this, and this Missouri 17 plant produces an ear like this. Not very impressive. But if you cross B73 to Missouri 17, so you cross this by this, and take those hybrid seeds, the so-called F1 seeds produced from that cross, and plant those seeds, they produce a plant that looks like this, and an ear that looks like this. This is what we call hybrid vigor. And of course, this is a pretty impressive plant and produces a very large seed yield. And what's impressive about this hybrid is not just that it produces a large ear of corn, but it's more resistant to things like drought and the wind and other sorts of things. And what's also very impressive about hybrids is that they are genetically uniform. We have here two uniform parents, and they produce absolutely uniform offspring, hybrid offspring, but uniform. And so we take for granted as we drive past acres and acres of corn throughout the Midwest or throughout Lancaster County that all the corn is the same height. But I'm constantly amazed by this simply because corn doesn't have to look that way. It is remarkably uniform, and it's remarkably uniform because it's all the same thing. It's all a hybrid produced from the same parents, and those parents were genetically uniform. And of course, this has important advantages for the farmer because now the farmer can go in and harvest that corn all at once because it will all be the same stage. You can basically till it all at once because the plants will all be the same height. He can basically treat it as one thing because it is, in fact, one thing. It's the same thing. Okay, so hybrid corn really, aside from producing a major increase in corn production, had another major impact, which is that it made it apparent that, that breeding, directed breeding, and use of and genetic knowledge could have a significant impact on agricultural productivity. In fact, the hybrid seed industry, the seed industry grew out of the production of hybrid corn because farmers had to buy their seeds. So that's one important thing that had a huge economic impact. But it had a huge scientific impact because it, I think, for the first time revealed that, that genetic knowledge was useful to agriculture. And it resulted in, it helped provide the inspiration for a lot of scientific research that then went on in agriculture to improve various kinds of, not just corn, but other crops as well. Where did the genetic variation that any kind of improvement depends on come from? Well, it all came initially from natural variation. That is, mutations or genetic variation that was present in the existing material that farmers had been saving over thousands of years. So if you just look at corn, here are just some ears of corn from South America and Middle America. It's incredibly diverse. You can have corn that looks like our Midwestern dent that I just showed you. You can have tiny ears of popcorn. You can have large kernels of couscous corn, and so on and so forth. You can even have corn that's covered where every kernel is covered by little tiny leaves. There's huge diversity in corn, as there is in any open pollinated or semi-wild, if you will, species. Now, some of the useful genetic variation that we've taken advantage of is not complicated variation at all. It's the simple sort of single gene traits that we learn about in high school that segregate 3 to 1, that segregate just like Mendel's colored peas, for example. So for example, here are 2 mutations. They are simple recessive traits, sugary 2, which are these sort of kernels here, and shrunken 2, which are these very ugly shrunken kernels here. And these 2 mutations are the basis for sweet corn. So when you eat sweet corn, what you're really eating are plants that are homozygous for this mutation or homozygous for this mutation and may have 1 or 2 or 3 other mutations that modify those effects. These are simple traits. Now, why do they produce sweet corn? They produce sweet corn because these are enzymes involved in the transformation of sugar into starch. So a mature kernel of corn is pretty tasteless because it's basically pure starch. You get corn starch, you're basically taking a kernel, and a mature kernel, and grinding it up. The starch in a kernel of corn comes from sugar that have been put together in various ways to make starch. If you block the enzymes that do that, then instead of making starch, you end up with the sugar, which is, of course, what the precursor of the starch was. And, of course, the ear, as a result, is quite sweet. Okay, it's just a mutation, simple mutations. So some of the natural genetic variation that we've taken advantage of are simple traits like these. Another example, this is not corn now, but rice. Perhaps the most important mutation in the history of humankind is this mutation right here, the semi-dwarf mutation in rice, because it's the basis for the Green Revolution that occurred in the late 60s and 70s that resulted in dramatic increases in rice yields in Southeast Asia. And similar mutations were important in the increase in wheat yields that occurred in Mexico and in Pakistan and India. They're all basically derived from these kinds of mutations. And these dwarfing mutations, very simple Mendelian traits, were valuable because they allowed rice or wheat to be resistant to this phenomenon called lodging. So the traditional varieties of rice and wheat grew tall, and if you added fertilizer to them, they grew even taller. And that made them susceptible to rainstorms or typhoons or hurricanes that would come in and blow over these tall fields of rice. But if the rice or wheat was short and had a strong stem, then these rainstorms or windstorms would not cause lodging and that you would get a crop out of it. So again, a very simple genetic change had a huge impact on agriculture. But much of the genetic variation that breeders take advantage of in the improvement of a crop is not something simple like a single gene mutation. It's what's called quantitative genetic variation, which is nicely illustrated by these set of corn ears that range from pretty close to white to quite dark yellow. This is quantitative variation, and it's controlled or it's the result of many different genes working in this case to control the production of beta-carotene, which is what gives the kernel that yellow color. So when we now look at this graph, we can say, okay, this increase in yield that occurred right here was a consequence of a new type of technology, basically taking inbreds and making hybrids out of them. But what happened here? Why did this graph continue to increase in yield? From this point on, essentially 100% of the corn in the United States was hybrids. So it wasn't just that people were adopting hybrids. They'd pretty much been completely adopted by around 1940. It was a very, very rapid transformation. All of this improvement was the production of better hybrids, okay? And it was basically breeders taking advantage of the natural genetic variation that existed in corn and using that natural variation to produce better plants. So what kinds of methods were used in this plant improvement? This is a sort of plant improvement component of breeding. Well, there are two general ways that breeding is accomplished. The traditional way is termed mass selection. This is the sort of approach that's been used for thousands of years. Ever since humans started growing plants or animals, they were selecting from a great diversity of organisms the ones they liked the best. Okay, and that's called mass selection. And this is not really based on any type of genetics. It's based on the phenotype. It's based on what the genes do to the organisms, what they look like. Okay? So farmers will say, oh, I like that corn plant, I like that sheep or whatever, and select it from a large number of other organisms, and then breed that animal or that plant with other ones or save the seeds because they assume, and typically this is correct, that the offspring of that plant or animal will share or have some of those traits. So it's entirely phenotype-based. More modern methods, and this is where genomics comes in, are not based so much on phenotype, though of course phenotype is extremely important. They're basically gene-based technologies. They're based on an understanding of the genetic basis for the trait one is interested in. And so what farmers do, or what breeders do now is take advantage of this information about what the underlying genetic basis for a trait is, and knowing that, they can select directly for those genes or for traits or for markers that are associated with those genes, and then use that in their breeding program. So here's an example of what you get when you try to define the genetic basis for a particular trait. So this is a fairly recent study published in Nature Genetics, and this group was interested in the genes that control resistance to southern corn leaf bite, a very damaging fungal disease. And so they took varieties of corn and they intercrossed them and mapped the genes, that is, determined the chromosomal location of the genes that controlled this trait using high throughput technologies that allowed them to look at all the genes in the genome all at once. Okay? And so these are sort of genomic technologies. And having information about whether the plants were resistant or not resistant to southern cornleaf blight, and knowing what the genetic composition of those plants was at every place in the genome, they could then identify certain sites within the genome that controlled sensitivity or resistance to that particular fungus. So again, this is an example of an experiment that was dependent on being able to look at all the genes in the genome all at once using various genomic technologies. Now, as you can imagine, if you wanted to now take advantage of this information and incorporate it in a breeding program, you wanted to make a plant that was resistant to southern cornleaf blight, you would have to worry not just about a single gene perhaps, but about multiple genes. So now we have this problem of how do you follow many, many, many genes in many, many, many different plants in order to identify those individuals that have the particular genes that control the trait that you're interested in. And so this becomes a numbers game. It requires then the ability to determine the genetic composition, the genotype of individual seeds, save the seeds so that if that seed has the genotype you're interested in, you can propagate it, okay? But be able to correlate then the seed with its genotype at an enormously large scale. And this required the invention of new technology, new engineering. This is the gene shipper. I think it was first developed by Monsanto. And what this very complicated looking machine does is basically remove a small bit of tissue from a seed, like a corn seed, and take that tissue, use it to determine the genetic composition of that seed, and then deposit the seed in a little plate here with a known address, so that the genetic information from that seed can then be correlated with that particular seed, and a breeder can then go back and use that seed. Now, if you look carefully at this seed, you'll notice, remarkably, that the chip has been taken out of exactly the same location on every seed. Right? It's not randomly chipped. This, of course, is fascinating because this required another bit of engineering, because these seeds are not fed into the machine in that orientation. They're fed in randomly. They're picked up by a little vacuum tube here, and a video apparatus determines the orientation of the seed because it has in its memory knowledge of what the morphology of that seed is. And it rotates the seed such that the endosperm, which is the part of the seed that doesn't contain the embryo and is not necessary for the viability of the seed, is brought up against a little saw here, and a little chip is removed from this seed, and then that's what's used for analysis. This is essential, otherwise you could kill the embryo by doing this. So this approach can be used for corn seeds, or it can be used in this case for soybean seeds, and for a variety of other different crops, once the computer knows what the seed looks like, so it knows which part of the seed to save. So that's genomics in corn agriculture. We'll get back to transgenics. We'll get to transgenics in a little bit, but before we do that, I want to talk about the other subject of this talk, which is cows. So if you drive again through Wisconsin, or even again through Lancaster, many parts of the country, and you pass a dairy farm, chances are you'll see these black and white cows in the field, and these are Holsteins. So the dairy industry is based almost entirely on this breed of cow, the Holstein. And that's because that's not the only breed of cow they could choose. Of course, they could choose a Jersey or a Brown Swiss or any of these other breeds of cow. But the reasons the Holsteins are the most popular cow for dairy production is they produce a lot of milk. Okay? There's genetic variation in cows for how much milk they produce, and also, it turns out, for the constitution of that milk. So that, for example, Jersey cows they don't produce as much milk as Holsteins, but their milk is higher in fat, for example, so they're good for making butter. Okay? But if you want milk, you use a Holstein. So how much milk do they produce? It is quite frankly remarkable. So typically, a typical Holstein will produce about 9 gallons of milk a day. And she has a lifetime production of about so-called 3.2 lactations, which is about 3.2 years. So basically, a cow is cycled. It's important to understand that cows only give milk if they've been pregnant. So basically, this is 3 calves and 3 years of production. And after that, her milk production drops, and she's basically turned into hamburgers. Now, to give you some sense of how much milk a cow is capable of producing, here's the all-time leader, Miranda Oscar Lucinda. She produced 28 gallons of milk per day. It's astonishing to think of how much milk that is. Okay. Now, it was not always so. If you look at milk produced per cow in the early 20s and 30s, it wasn't nearly that much. You know, here it's this is milk per cow in pounds. You know, typically milk is measured not in gallons but in pounds. So 4,000 pounds per year. And then sometime in the 1950s, things changed dramatically. That change, that increase in milk production, again, had two sources. Part of it was genetic. Part of it was breeding better Holsteins. And part of it was actually also so-called phenotypically, is what the cows were fed. It was better nutrition and it's the technology, if you will, of milk production. But I want to focus on the genetic component of this in particular. So to give you some sense of this, let's look at what a typical farm would have looked like back in the 20s or 30s. And we don't go to the United States for that. We can go to Africa, to the farm of this woman here, Purity Nagai. So her farm is located in Kenya, not far outside of Nairobi. And Purity has 3 cows, and they're Holsteins. And they're Holsteins for the same reason that farmers in Wisconsin have Holsteins, which is they produce the most milk. But these Holsteins are not high-bred Holsteins. They are sort of run-of-the-mill Holsteins, and they don't eat diet that's been chemically modified and highly regulated. They eat basically cut up corn stalks. Okay? And as a consequence of that, instead of producing 9 gallons of milk a day, they produce 1.8 gallons of milk per cow per day. Okay? Or about 564 gallons per year. Now, because we're talking about Kenya here and not the United States, there is in fact no infrastructure for keeping this milk cold. So the milk has to be sold immediately. It's kept unrefrigerated, sold to a middleman, and sold at the local market. But remarkably, nevertheless, those 3 cows are Purity's main source of cash. Very low-yielding cows, but nevertheless important for her livelihood. Okay, so what is it? What happened to take a cow like Purity's cows, which is presumably much the way cows were maintained at the early part of the 20th century, to generate this dramatic increase in milk production that we have become accustomed to today? Well, until very recently, this major increase was basically sophisticated mass selection. It was, just as in the case of corn, it was going and finding the best cow for a particular purpose. But it was sophisticated mass selection, because breeding is a numbers game. The more plants or animals you look at, the higher the chance that you'll find a high-yielding one and can select that from the mass of other organisms. And farmers do not anymore milk cows in this way, okay? Which does not, of course, very labor-intensive. And not only that, but it doesn't provide a lot of information about the yield of that particular cow. Nowadays, of course, all cows are milked by machines. And the machines that milk these cows keep a very accurate record of how much milk the cow produced. So we now have a lot of numbers, a lot of data for the production of each of those cows. Not only that, but we actually know who the parents of this cow are, because this cow is very likely the daughter of a previous cow owned by this dairy farmer, and she is the product of an artificial insemination. So almost all dairy farmers do not allow their cows to become pregnant the old-fashioned way. They control that process by choosing sperm, from a known bull who has been proven or shown to have high-yielding daughters. So the child's productivity is known, and her father and her mother are known. So we have a lot of information about this particular cow. And that means that you can identify which are the best fathers and which are the best mothers. So here are some examples. Here are 2 very important bulls, round oak rag apple elevation. And he had 70,000 offspring because his semen was diluted and then sent around the world. Whoever wanted to pay for it could get it. And one of his sons was a bull named Hanover Hill Starbuck. And this was an amazing bull. He had $20,000, and he had 685,000 doses of semen that were sold. That semen was worth $25 million. Okay. So farmers could buy a straw of that semen if they wanted to basically have a calf produced from semen. He was so valuable that in 2000 he was cloned. So 2 months before he died, they harvested fibroblasts from him, and they turned those fibroblasts. They 68 nuclear transfers, and one of them survived, and here's Starbuck 2, a clone of Starbuck 1. Now the interesting thing, this was done in Canada. Now the interesting thing was that Starbuck 2 was kept alive for 10 years, and it turned out Canadian regulations did not allow cloned animals to be used for this purpose, so in the end they sacrificed them after 10 years because it was just expensive to keep them around when they weren't basically making any money off of them. That's the male side of reproduction. But of course, we need to know about the female side as well. And the female side is just as interesting as the male side. So here are 3 cows from Gloryland Farms. As you can see here, these are very valuable cows. This one sold for $100,000, and this one sold for $410,000. And they're related to this animal right here, Gloryland Lanneray. Her offspring are highly valuable. So valuable that when Lanneray went down and got sick, the farmer who owned her brought in a chiropractor to massage her and keep her happy, and brought in an acupuncturist as well to keep her in feeling in great shapes, because he was interested in getting her eggs. He needed, because he wanted her DNA, and he wanted her DNA for as long as possible, or more appropriately, not just her DNA, but the eggs that she produced, because of course when inseminated with the right male, those eggs were worth a lot of money. More specifically, the calves that they produced were worth a lot of money. So that kind of breeding is old-fashioned breeding. That's mass selection, right? Those cows are valuable because they typically produced high-yielding offspring, and so therefore the assumption was that, okay, if their offspring tend to be high-yielding, then we should keep them around. And so those cows were valuable in the same way the large ear of corn is valuable, because of the phenotype of daughters. But that's a prediction, right? It's a numbers game. And genomics has changed that. Because now, in the same way that you could identify the genes that made a corn plant resistant to some fungal disease or improved yield, one can map the genes that are important for milk production or for any other trait. And so no longer do you have to hope that the children of a Starbuck or of a Lanneray are going to be high yielding. You can go in and determine their genetic composition, their genotype, and determine if they have the right genes for whatever trait you're interested in, and then screen their offspring for the presence of that particular gene, and then choose those offspring that have that gene. For your purposes, these animals right here may have come from a very expensive male, but are potentially useless because they don't have the right genetic composition, even though their parent may have been high yielding, for example. How is this done? Well, it's done using these sort of microarrays or chips where you have on this little slide pieces of DNA that represent sites across the entire genome. And then you can take DNA from those animals and hybridize it to this little chip, and it will tell you whether or not the animal has the particular gene that you're interested in or not. There are, of course, companies that will do this sort of test for you and do other very high-tech tricks. So here's one of them, TransOVA. It will do embryo transfer, so it will take embryos from super-ovulated cows like Lanneray that have been treated with hormones to produce lots of eggs. She's inseminated. Those embryos are removed, and they are then transferred to so-called recipients or surrogate mothers, and calves are raised to maturity, and this company will do that. They'll do in vitro fertilization, sexed semen. So, of course, 50%. If you're a dairy farmer, a male calf is useless to you. In fact, that's where our veal comes from. It basically comes from male calves that are useless from a milk production perspective. But of course, if you don't want to get rid of them, that's expensive, you can sex your semen, and so only use semen that don't have a Y chromosome, basically. And other sorts of high, even up to cloning, these companies will perform it for you. So milk production, dairy farming can be a very high-tech proposition. But of course, perhaps the most sophisticated sort of modification that scientists do these days, that breeders do these days, is called transgenesis, making what we now typically call genetically modified organisms, although in reality, everything we eat has been genetically modified over 10,000 years of human cultural evolution. So when we think of GMOs, as they're typically called, we typically think of organisms that have had some foreign gene inserted in their genome. The classic example, the most dramatic example, certainly is the green fluorescent protein, which originally came from a jellyfish. So here's a tobacco plant expressing green fluorescent protein, and here are some mice that are expressing this protein. And these, of course, are dramatic because they're brightly green and fluorescent. Now, there are no transgenic animals used as food sources today. So if you're concerned about transgenic animals, you don't need to be, because there are none that are as a food source. But we have lots and lots of, we don't have lots of different species of transgenic plants, but we have a lot of transgenic plant material in our diet. So how are transgenic plants created? They're created primarily, although not entirely, using this bacterium, Agrobacterium tumefaciens. Agrobacterium tumefaciens is a naturally occurring bacterium that's the basis for this disease called the crown gall disease. So if you walk around the forest and see trees that have these burls on them, these outgrowths, irregular outgrowths, these are typically caused by the infection of the cells of that tree with this bacterium. What the bacterium does naturally as part of its life cycle, so this is a picture from my colleague Andy Binns, is the bacterium attaches to the plant cell wall. So here it's basically, if you will, mating with or infecting a plant cell. And that attachment then allows the bacterium to transfer a piece of DNA from its chromosome into the plant cell, where that piece of DNA then becomes incorporated in the DNA of the plant. This is a transfer of DNA from a bacterium into a plant. So a transformation event that occurs naturally as part of this bacterium's life cycle. Why does the bacterium do this? Because on that piece of DNA are genes that cause cells to divide irregularly, as well as genes that provide the bacterium with a food source, with an amino acid that it uses as a food source. So basically the bacterium is making lunch for itself by doing this transformation. But of course, if you want to use this bacterium to make a transgenic plant, you don't want a tumor. You want cells that can be transformed back into plants. And so what scientists have done is they've removed from this piece of DNA the genes that cause cell division and replaced them with genes of whatever trait they want, including something like green fluorescent protein, which is shown here. This is the roots that are transformed by Agrobacterium. So what kinds of genes have been put into plants? Well, here's one of the most common ones. This is a gene from the bacterium called Bacillus thuringiensis, the so-called Cry1 protein, which is a protein that is toxic to Lepidopteran larvae. And plants that are transformed with this gene, that express it, are basically kill the larvae that try to eat them. And so you have here nice, healthy-looking ears instead of these very damaged ears. Or this is also toxic to European corn borer, for example, or other corn borers, for example. So much of our corn has this particular gene in it. Another trait that has been introduced into plants is resistance to herbicides, so-called Roundup resistance, that allows farmers to spray their fields with this herbicide because the plants are resistant to it. So GM organisms, corn and soybeans and cotton, are the three major crops that are grown in the United States that are transgenic, are now a very important part of, important in the sense that they're grown a lot of our crops. So this is in 2011. 94% of the soybean crop was resistant to Roundup. Okay, so if you're eating soybeans, chances are they're transgenic. And here are the numbers for some other crops like cotton and corn. And so about 70% or so of these other crops are transgenic. And they can contain either, in this case, this is herbicide tolerance. So these soybeans contain a gene that makes them resistant to an herbicide. Or they can contain something like Bt, which then confers resistance to insects. Now, it's not just a single transgene that is necessarily in this plant. So this is a page out of a hybrid seed company that I got actually at the Pennsylvania Farm Show. And so here's one variety of corn, for example, called Genuity Smart Stack. Okay? And this particular variety of corn has 7 different transgenes in it. It has 2 transgenes for resistance to Roundup Ready 2, excuse me, 1 transgene for resistance to Roundup Ready. It has Liberty Link, which is another herbicide, so it's resistant to 2 different herbicides. It has 3 genes for resistance to corn borer and 2 genes for resistance to rootworm. So that one variety of corn has 1, 2, 5, 7 different transgenes in it. Now, of course, this technology has not been greeted with open arms by everybody. There's been a lot of resistance to it in some parts of the world, on Greenpeace and in other parts of the world, primarily because, for a variety of reasons, some of it have to do with the fact that people object to the fact that large companies like Monsanto own this material. So people are opposed to it because they don't like the idea of food being owned by companies. But most people object to it because there's some deep-set dissatisfaction or nervousness about the technology that has been used to put genes into plants or into animals. Humans have never encountered this technology before. It's not something that the person on the street knows much about, and so naturally people can be quite nervous about it. And another reason for some of the nervousness is that, in fact, the traits that have been put into these plants, like resistance to herbicides or Bt, from the standpoint of our perspective, what value is that to us? You know, we don't personally benefit from it. The farmer benefits from it. The farmer can spray less herbicides on their crop or less pesticides on their crop, but we don't see that. But of course, this technology can also be used to put interesting and potentially useful and nutritious sorts of traits into plants. This is a very interesting example. It's a so-called golden rice that's been engineered to have high levels of beta-carotene. Okay? This is not done by a company, done by a scientist in Belgium. And in fact, the idea here was to make a rice that was high in vitamin A, to basically make a yellow version of rice like we have yellow corn. Now, some people have objected to this because they say, well, you know, you're putting beta-carotene into rice and we don't know what that's going to do to humans. There are lots of arguments that one might use against it, but that's a specious argument. Because we eat carrots quite happily because they're supposed to preserve our eyesight or what have you. And so, carrots are not naturally orange. If we were worried about high levels of beta-carotene, we should only eat white corn because yellow corn has got lots of beta-carotene. So the argument that putting too much carotene into rice might be bad for you just doesn't make sense because we typically eat crops for this purpose. Another example of how GM technology could potentially be useful is the so-called Enviro Pig. So this pig it's a GM pig, and it was created for the specific purpose of reducing phosphorus pollution. Okay? And basically they're transformed with a gene from bacteria that allows them to consume this particular compound and reduce the amount of phosphorus in their urine and manure by 65%, because phosphorus contamination is a very serious problem. But this pig is not available now because of the technology that was used to create it. Not because of the trait. The trait is a useful trait. It reduces pollution. But the objection was to the means by which this pig was produced. So we are at an interesting, it's an interesting dilemma that we face all the time. Consider the cell phone, the smartphone. The smartphone allows you to communicate with people, to get information off the web, to check your email, to do all sorts of useful things. But of course, it also allows you to check your email while you're driving, or communicate with your friends while you're crossing the streets, and has resulted in an increase in the car crashes and people being killed by cars. Right? So the apparent, innocuous smartphone has a good side to it and it has a negative side to it. And we as humans can choose. Do we want to take the risks of dying in a car crash for the benefits that it might provide us. The same thing is true of any new technology. We can decide now, when confronted with GM crops, do we want to take the risk of some potential problem, which may exist, we don't necessarily know that it exists, or eliminate it altogether to avoid that problem and potentially take advantage of some of the benefits that it can provide us with. This is the decision, the options that we're provided with, and it will be interesting to see where humanity, where we in America or in Europe finally come down on this issue.